Our research basically is on the cell biomechanics research, and we look at how cell responds to mechanical loading. The reason that we're looking at cell biomechanics in our body is that in our body, the cells are not in just a static condition. They're actually exposed to all kinds of mechanical stimuli, pressure, shear stress, compression, stretch. So all these dynamic and mechanical stimuli are actually critical for cell function, for cell metabolism. Therefore, if we can understand better how cells live in such a dynamic mechanical environment, we can come up with a better strategy to treat diseases.
So my research is focusing on developing a new device which can capture the circulating clonal plasma cells in the blood of the patients with multiple myeloma. When these myeloma cells leave the bone marrow and enter the bloodstream, the patient may be at an increased mortality risk. However, these circulating multiple myeloma cells are difficult to detect, especially at low levels. So I am conducting research with our hospital collaborators. Firstly, use a computer model to design a microfluidic device containing tiny pillars that can isolate the circulating myeloma cells from the small samples of the blood.
My research focuses on building a bone metastases on chip system, and we want to see the microenvironment of bone metastases. And with this microfluidic device, we can mimic the microenvironment of bone metastases, and then we can regulate osteocyte under mechanical stress, and then we want to see how it affects the breast cancer exacerbation. And it's important because nowadays bone metastasis is really a serious problem for breast cancer patients, and they really suffer a lot. And with this investigation, we might come up with some insight of how the osteocyte regulate in bone microenvironment for the exacerbation of breast cancer.
My interest is in developing new microfluid devices for investigating bone mechanobiology, studying how bone cells react to different kind of mechanical stimuli. And one of the limitations in the field right now is that we're still relying on very standard culture wear devices, whereas the capability of microfluidic devices hasn't been utilized for bone cells. So in my specific research, I'm interested in developing a platform we can study the response of these bone cells to different levels of mechanical loading, all within the same chip. So we can load a whole population of cells with the gradient of different type of forces and look at their response. And furthermore, also study how they communicate that response to other types of bone cells in order for it to orchestrate sort of the whole bone tissue. So we're sort of building from the ground up sort of a bone on a chip concept. And hopefully in the future, we can slowly incorporate 3D architectures into these kind of devices, sort of build a 3D in vitro model of a bone tissue.